Good morning, ladies and gentlemen. And I'm very happy to be here at 2022 BIOPLUS-INTERPHEX Korea and welcome you all. I am Bae-jin Gon, moderating today's session, TPD and TPU session. And actually, Scientific Intelligence Office is my work that I like the most. And personally, to the other company, Scientific Intelligence Advisor is the role that I am playing. And when you read my article in Scientific Intelligence, writer is what I am doing and serving as. And actually, I have prepared for 2 slides. The first slide is about protalk. And actually, the word protalk is falling under the category of trademark of a company, but many people refer this technology as protalk. So when you return home, and if you want to do study and survey, so protalk, actually you need to study on the possibility of protalk's potential. And actually, I wrote this article 4 years ago. And also, 환경 바이오 news was something that I wrote for, and I wrote an article titled EGFR treatment can be all trickled down into the protalk and the future look like of next generation protalk, and also the grand analysis on the B4 companies related to protalk listed on Nasdaq is the article title. And for today's session on July, I wrote an article on that day. And as a disclaimer, before I kick off this session, protalk and antibody conjugate, or ADC, have shared the tripart structure. So to my surprise, on April 9, Daiichi Sankyo announced that the jury delivered the verdict on ADC HER2 specific ADC, and HER2 violates the patent of Seagen. And this patent strategic war might be transpiring in the future warfare of protalk, and domestic companies should overcome these barriers in the protalk warfare. So Korean companies for protalk as a latecomer have to think about how to secure the patent of E3 ligase, linker, and ligand. So actually, for our first presenter, we have In Young-soo, CEO from Prager Therapeutics, and he will deliver on a novel platform technology to develop protein degrades. And Prager is very interesting to me. Do you know what the Prager means? Protein eraser. So CEO In will deliver his remarks and presentation. Thank you.
First of all, thank you very much for inviting me to this very special occasion. And thank you very much for the organizer. And I'd like to extend my appreciation to Dr. Bae-jin Gon, who will serve as a moderator. And as introduced, I'm the CEO of Fraser Therapeutics. My name is In Kyung-soo. And as you can see in the title of the slide, I would like to introduce the platform technology, which is called as SPIDM. The topic I would like to cover today is first, I will introduce the company. Then I will also talk about the TPD briefly because other speakers will also cover this topic and why we need to have new TPD. Then I would like to talk about the SPIDM we use. What are the characteristics of this technology? This is the overview of the company. In 2019 October, our company was established, so it's been about 2.5 years, and our employees are 25, and about 18 people are researchers, and about 8 people have PhD. And early last year, we finished the Series A funding. So when it comes to the TPD, many people have interest. And as you may know, this is about target protein degradation. So how we can induce the target protein degradation? Basically, we need to use the mechanism of the cells. So in the cells, there are a protein-dependent degradation pathway and lysosome-dependent degradation pathway. So based on UPS system, soluble proteins will be degraded, and organoid and membrane will be degraded based on the lysosome dependent pathway. So UPS dependent pathway will be used for some companies, and autotoc and autotoc lysosome-dependent degradation technologies have been developed by Korean companies as well. So the topic I would like to focus now is how we can modulate these two pathways. So this ubiquitin is very important process, and K48 and K29 linked polyubiquitination will induce this process, and endocytosis or autophagy will create the K16 linked polyubiquitination. So there are two different pathways and two different way of ubiquitination, and depending on the combination, the pathway will be determined. So I believe you are well aware of this process. So I simplify the process. This is a protein of interest or target ligand, and the E3 ligand is there, and it will be connected with a linker. So when you look at the mechanism of action, then molecules will be combined with the E3. And here the important point is the tunneling complex. There should be tunneling complex. So this proximity will be induced. Then ubiquitin will be delivered to the protein of interest. Then this protein of interest will be degraded. That's what we call the protoc. We have a full house here, and as you may know, the field of TPD is getting attention. And as you can see in this slide, there are a lot of deals taking place. And even after I made this slide, new deals are coming up. So big pharmaceutical companies and many biotech companies are having a lot of interactions. When we look at these, and when we have meetings already, big pharmaceutical companies are jumping into the bandwagon of protoc. And top pharmaceutical companies have 20% of the pipeline in the degradation-related field. So indeed, I would like to talk about the potential or excellence of protoc. I believe other speakers will also cover this. And I would like to also talk about the challenges we realized. In the field, many people talk about the PK profile. When we create the protoc, the molecule is, in many cases, very small, 700, 800, or even 1,000. So the PK profile is really different from other drugs. So simply say, due to the big molecular weight, there are limitations in absorption, and it can be compared with other drugs. So the intracellular delivery is not happening frequently. And the second challenge we realized is E3 ubiquitin ligase. And companies are now developing new E3 ubiquitin. I believe at the end of this year, there will be many clinical studies. And aside from one or two undisclosed studies, many are using these E3 ubiquitin ligase. And there are limited number of tissues. So there are limitations in selecting the targets. And I believe you are well aware of the situation in the field. The structure is very difficult to understand. So there are limitations. And when it comes to the CRBN, expression profile is good, but there are pattern barriers. This is a big issue. And indeed, when it comes to the E3 ligase, we need to use a specific ligase, and there might be limitations. These are the challenges we thought. So in our operation, we decided to change the direction. So that's why we came up with the technology called the SPEEDM. So SPEEDM is a selective protein degradation enabling moiety, and it is using different mechanism compared to PROTAC. So I'd like to introduce the mechanism of action. Basically, we use the ubiquitin liquiding moiety and target binding moiety. And as you can see at the bottom of the slide, instead of inducing the ubiquitination of the target protein, it is directly receiving the ubiquitination. So in our opinion, I believe this is the only chemical ubiquitination we developed. Basically, it is combined with the protein of interest, and this URM structure is receiving and hijacking the ubiquitin from the E3. So there is an interaction chemically, and it will be degraded after all. In the initial phase, we had doubts whether it will work, but we realized that chemical interaction is happening. So we created the ubiquitination from the pure state. And this is not the protein, rather we created a new structure and we added the E3 and induced the ubiquitination. And finally, we pulled down this and confirmed the outcome. And we used lots of E3 ligases, and we can see that they are making and inducing good ubiquitination. And this is not the biotin. We targeted the biotin, and we combined this with ubiquitin. So after the combination, we were able to see 580 ubiquitin combined together. So we analyzed lots of patterns of ubiquitination, and we were able to confirm different situations here. So the biggest benefit here is we can induce the ubiquitination depending on the situation. K63 dependent ubiquitination can be induced, then we can access to the aggregated protein that is not possible in the autophagy. And here, the target protein is decreasing, and A3 band is decreasing. So we confirm this outcome, and autophagy is activated. And when it comes to the membrane protein, we used the two types of SPEEDM, and a ligation was induced successfully. So these have different mechanism of action. So we secured different TPD, and the question is whether target selectivity can be secured. So this is an example we would like to share. This is a P3A mechanism, and target binder has been already developed, and we shifted it into sputum. So a consentive negative form P3 can be degraded very well. But when it comes to the dead mutant, it is not degraded. So we were able to see this pattern. And as for the tau, aggregated tau is well degraded by the sputum we developed. So indeed, the target binder is requiring specificity, and without changing the specificity, we were able to maintain the specificity, and we can also induce this outcome. Basically, compared to the conventional protein, what is different? Basically, the size of the sputum is much smaller than the conventional one. So PK profile is much more improved. When you look at the slide, of course, we will use much more rigid linker in the clinical field, but facial ligand will be used as a linker in the product. And when we look at this, at once we can realize that this is a product. However, for the sputum, we can disclose the full structure. Then, without the background knowledge, it is difficult whether it is TPD. So the structure is very simple. In the development phase, you may have the experience. This linker is very important. So when we do the optimization, the linker can be longer or smaller, and the orientation can be changed. Then activity will disappear. So there are many challenges we experienced. But as we said, we have different mechanism of action, so the optimization process is much more simpler. As I mentioned before, this is the early version, and as for the P38, we created the product version and sputum version, and we compared the PK profiles. And when we compared the bioavailability, it was higher in the sputum. So overall, the PK profile has been improved, and in vivo profile also has been improved. And we were inspired by the outcome because in the brain area, we measured the concentration, and the BBB was almost 65%. And as for the counterpart, the level was only 35%. So the size was smaller, and the PK profile has been improved. So now it is even possible to achieve the BBB. And we also used a wild-type mouse and injected with IV. And in the brain tissue, in all three mouse models, we were able to achieve the post-preparation and the ligation. And we also achieved the reduced basal level activation. So we believe that this modality will work. So we have been optimized this system, and we combined many things. Now, overall, we have 4 types, and there are many derivatives. So based on these libraries, we will expand this with lots of pipelines. As you can see, most of them are related to the CNS, and the oncology program is also there. When we implement this program, we need to see the pattern. And as for the BRD4, we can compare this with the PROTAC. So BRD4 is a common target. And according to the dose dependency, we can see the outcome. And we also detected the activation. And when we administered this at the cell level, we were able to see the BRD4 dependent ratio, and it has been decreased. And the efficacy has been also improved. And we have another platform. Another platform is Tau Speedem, and this is the first screening data. As you can see in the screen, there are different candidates, and phospho-tau has been removed in the outcome. And in the in vivo model, we used a mouse that expressed the human mutant, and two types of sputum have been selected. And in the in vivo system, we were able to see good degradation level. So in conclusion, basically, when it comes to the sputum, instead of using specific ligase, we use authentic E3 ligase, and we can adapt to the situation. And membrane protein or aggregated protein can be the application. So we have the strength here. And compared to the existing product, as for the CRBM, it is widely spread in serum, but cellular localization of human body are all different, and the concentration of the membrane can be different. So they are located in different places. So creating the ternary complex is difficult. Some targets work well, but the others does not work. Compared to this, our platform can be more versatile, we believe. And as I said before, drug likeness has been improved. So PROTAC had many limitations. So in the US, many companies are moving on, but molecular glue is a good platform, but as you know, with modification, we can design it so that it can degrade a specific target, but it is difficult. But when we use SPIDM, we can design it as we will, and we can degrade a specific protein. So we combine the benefits of the molecular glue and PROTAC. I made a presentation really quickly, but I have one question. How many URM do you use? In large, we have 4 URM, and if we count everything, then it will be more than 100 URMs. Thank you very much for the presentation. My name is Yu Hae-dong from UnoQ, and this is new concept of modality. It was very interesting presentation. My question is, as you mentioned, URM has 580. The overall SPIDM is targeting, and that SPIDM has 560 URM, but usually it is 150. I think it is my misunderstanding. I thought that the URM itself was 587. So even if we add the linker, then we can have a PROTAC and URM, so the size will be similar. Then in the PK profile, I thought that there will not be advantage, but if the URM itself is smaller than that, so the 560 is overall molecular weight and not the URM. And in the spirum, Did you observe any hook effect? We had about 10 targets, and we've been experimented several times. And in most of the times, we were not able to see the hook effect, but sometimes there were minor hook effect. But basically, we believe that there is no hook effect. I'm from TeraFux. It is not ubiquitinated to the POI. So what if it is separated from the POI? So we need to see the target binder and target interaction, and it will be depending on the system. The ubiquitin is not binded with the POI strongly enough. Theoretically, we believe so. But on data, degradation efficiency is not that low compared to the fine-tuned product. So we need to see the affinity with the target and ubiquitin affinity. So there is a specific range. But if the affinity is strong, then there will be more reaction. So when we bring the ubiquitin, the actual affinity is not that high. So it will be combined with the target. And once it is combined with the target, then the efficiency of the degradation is not that different. We'll take the last question. Thank you very much for the presentation. You mentioned about the hook effect. Basically, a ternary complex should be created. Then if there is no hook effect, then it is the ternary complex concept is not applied correctly. So hook effect is one of the criteria we explain. When it comes to the hook effect, we believe that in the process of creating the ternary complex, there are two ways of affinity. So one way is occupied already. So binary complex will be increased in one way. So once it is happening, it is supposed to be happening. There will be next step. So it can be separated, and then the process will be continued. So that's why we believe hook effect is not observed in many cases. But inside how it is actually processed, we need to see the affinity between the POI and the ubiquitin. So it is difficult to give you the definite answer. Physical property will be improved, then we will have benefits, but we will also have the demerits as well. So we'll move on to the next presentation. Thank you very much for your presentation.
And now we will listen to Optera CEO Choi Si-woo. Good morning, ladies and gentlemen. As introduced, I am Choi Si-woo from Optera. And previous speaker mentioned the TPD well. So actually, I will touch upon the lead program that we are developing right now. And actually, you will listen to more details. CEO In also mentioned we need to identify novel E3 ligase, which is in limelight globally. So what kind of strategy do we have to be equipped with is what something that I want to mention. So this is the slogan that we have pursued. So synthesize possibilities, degrade despair of patients. And based on TPD, we are developing first-in-class drugs. And we started this company with the 5 co-founders. Actually, they have accumulation of expertise in cell trion, and we have been established in 2018, and we have $43 billion through 3 rounds of series financing. And also, we have been certified Innobis. And from now on this year, we are developing business in earnest. And in April, we have signed an MOU and licensing out in collaboration agreement with Yuhan Pharma. And our company employed 57 workers, and also we have external personnel, and 37 are focusing on R&D members, and non-R&D members are 20, and other 20 have focused on study on protein and protein R&D. Is what we are doing, and building on the preclinical data, we should be quick to reflect that data into our future plan. And also 7 are in non-clinical team. And E3 identification and discovery is inviting bioinformatic teams, and quality check IPT and other development teams are also engaging. And over the larger concept, we have Protect technology, violent degrader, and license out is one business model we are pursuing, and other business model is platform business to identify novel E3L platform development. And there are 2 pillars. So actually, my presentation consists of 2 chapters. The first chapter is about lead program, and the other chapter is about what logic we have to build novel E3L platform. So the mechanism of action of Protect is well known to you, so I will skip that. And the functional benefits of Protect is well known to you. So in a nutshell, traditional small molecule inhibitors should be developed only for protein that possess binding pocket with active site. So it is very undegradable target. But however, Protect can degrade disease-causing protein by binding the protein in any manner. So undruggable targets can be degradable by Protect technology. This is one of the benefits that the Protect has. And the small molecules mechanism of action is based on inhibition. However, Protect is based on degradation. So it is about degradation of disease-causing protein fundamentally. And before the presentations and Q&A sessions, some people mentioned this. The creation of tunneling complex is important in Protect. So designing materials is something that we have to think. But however, the Protect can reduce the off-target issue and effects, and we can also improve the toxicity profile through this. And the big difference between the inhibitor and degradation, actually one drug inhibits one protein. So this is occupancy-driven mechanism of action. However, Protect can degrade multiple proteins. It is working as a catalytical effect like an enzyme. So it's event-driven mechanism of action. And as you can see from this presentation, the previous speaker also mentioned this. So briefly, the top 10 pharma companies have signed an agreement with TPD partners, and last year we see an increasing in technology deals. So Arvinas and Pfizer have pipeline license deal, and upfront is $700 billion and milestone $1.4 billion, and equity investment is about $350 million. And Vividion, which is in preclinical trial stage, and it is acquisition merged by Bayer, and also this is about 2.6 trillion won. So this is what how we can proceed R&D first. We proceed ID or target identification. So if we decide if there is a known binder which is applicable for a product, so there might be a lot of binder known to us because of the kinase inhibition. However, when we look at the undruggable target protein, we do not have a known binder. So if there is a known binder, we have carried out fast track discovery. Building on the existing binder, patent is important, and IP strategy should be reinforced. So we have to file a patent in a rapid manner. And as I mentioned before, when we do target undruggable proteins, especially novel E3L, this is no binder for this. So from the screening and to the in vivo POC, based on chemoproteomics, we have developed this in-house. And with the collaboration with external partners, AI virtual screening is utilized together. So when we identify one target, we have utilized a variety of screening tools to find the optimization binder tool. And in the pipeline internally, including lead program, we have 6. And 6 tasks is consisting of fast track and long-term track. And Noble E3L has 4 long-term track discovery programs. And now let me touch upon the lead program that we have. And actually this session, my presentation is cell cycle protein degradation. And the PLK1 is our POI. And PLK1 is the target protein. And building on this, we are developing the PLK1 degrader. And one of the PLK1 inhibitor and CRBM binder inducer are customized. And we link these two with customized linker. And these tasks, when it comes to PLK1, there is no approved PLK1 inhibitor or degrader currently, so we can overcome this barrier with our PLK1 degrader. Of course, I believe this will be a first-in-class drug, and material size is big, but when we do oral administration, 20% is witnessed, so we can consider the advantage of oral drug. And the one drug candidate, which is in clinical trial, compared to this competent with more potency. So our major indication is CSCLC. But PLK1 degrader that we are developing can be also having a lot of indications in different tumor types. The reason why we have SCLC as a major indicator, actually the 5 survival rate is less than 7%, and the survival duration is less than 1 year. This is the case when we do combination therapy of cancer therapy. So this is a very intractable disease. So when we develop a first-in-class drug for SCLC, then we can utilize the advantage of fast track of the regulation authority. So this can allow us to advance into the market in a rapid manner. And also we have carried out in vivo trials on a variety of types of tumor cells. And this is fast track program. So we have 3 composition of matter patents for PLK1 degrader, and we have 4 registered. And including other tests, we have a total of 60 patents filed, and we have more than 10 patents. And PLK1 is aiming to arrest the G2M phase of cell cycle. And PLK1 overexpresses in multiple cancers compared to the normal cells, and the prognosis is not that good. And there are a lot of attempts to develop PLK1 inhibitors, but this only to be failed. And onvansertinib is only candidate in clinical trial. So when it comes to inhibitor, as I mentioned before, PLK1 expression is going up at once, but it also going down. The activity of PLK1 is going down. So we have to dose escalation in accordance with this. But this leads to hematologic toxicity. But when we do degradation by protect, but at the same time, we can utilize the expression of PLK1 at a lower level. So we have advantage on mechanism of action, and also protect has a good selectivity. And there are a lot of issues in terms of on-target toxicity, but PLK1 can be lowered to 80 to 90%, but there is no neutropenia. So compared to the inhibitors, when we utilize degrader, we can control PLK1 expression. And also, along with these PLK1 tasks, we have CRISPR-Cas. We have utilized CRISPR-Cas9. We have live cell library, and we also establish cell line to be used for screening. So you can see the pattern here. Sorry for my mistake. And as you can see here, from the Western Brussels degradation, DC50 is a picomolar, and 90 is 3.3 nomolar. And we have identified the anti-cancer efficacy in SCLC. So PLK1 specifically degraded by proteomics. We can confirm this by proteomics. And among the proteomic family, there are a lot of proteins. PLK1 specific degradation can be happening. And in the industry, Servium neo-substituted issues are thrown. But GSPT1, Ecolage1, Ecolode iron loss, and their substrate are not degraded by PLK1 degrader. And onvansertinib is one candidate in clinical trial phase, and 140 cancer cell line are subjects of our panel assay, and most of cell lines compared to onvansertinib show the prosperity of PLK1 degrader compared to anti-cancer efficacy. And we also categorize a lot of cancer types. Onvansertinib has fluctuation, however, degrader has a plateau effect when it comes to anti-cancer efficacy. So in order for us to expand the indications, we have carried out additional experiments on 5 tumor cell type. And we have identified the antigen effect, and we have achieved complete response. So we are in the preclinical trial, and PK/PD profile are relatively good. And building on the data we have gathered, we try to global LO for these tasks with partners. Building on CDA, we have carried out meetings continuously. And another pillar of this model is novel E3 ligase platform or development. Novel E3 ligase discovery is something that we are pursuing, but we are not confined to this. CIFHR are not sufficient to cover all the target proteins. This is a global consensus. So compared to the two E3 ligase, we have to target and identify more optimized E3L or ligase. So we have utilized upgrade solution, which will be addressed later on. So we are focusing on what to develop. Basically, we utilize single cRNA sequence data, and when we have developed novel E3L, then we have to think and ponder upon how to develop. And covenant probe finding is what we are pursuing, and also we have external partnership on this issue. So upgrade solution is operate in this manner as follow. So cell cycle protein, a lot of inhibitors are developed, but toxicity is a limit, so they dropped off from the clinical trials. So low hematologic toxicity is a direction of our development of degrader. And single cRNA sequencing is utilized, and we see RNA expression at a single cell RNA level and at a protein level, and we gain insight. And IMS Chimera are utilizing analysis on PCR level, but our solution is analyzing target protein expression level at a single cell level. So this will lead to enhanced reliability and accuracy to discover promising novel E3L. And this is an example of upgrader solution, the disease data. And this slide and the slide and next slide contain the example of upgrader. And we try to verify RNA expression level of normal cell and patent-derived tissue. There's a certain target protein B, and we also verify RNA expression level of novel E3L in normal and patent-derived tissue. Is our E3LX compared to the CRN is overexpressed in patient-derived cell types. So protein B target together employing novel E3LX will be developed. So this kind of pairing will be working better than the other combination. So what I like to draw attention to is the E3 ligase 1. And there are a lot of blood cells, and there is a variety of expression patterns. But E3 ligase 1, actually E3 ligase 1 expression level is significantly lower in the blood cells compared to the other E3 ligase. So if there is a limiter which is very hard to be developed because of the higher hematology toxicity, we can utilize this to overcome this barrier. With this logic, we are identifying novel E3 ligase, and this is entire flow chart. As I mentioned before, we have novel E3L planning, and we utilize analysis of upgrader, and we discover promising novel E3L and study novel E3L characteristics, and novel binder screening will be done by Upbit and cancer specific, but at the same time, lower blood toxicity is now in Protect POC test and E3L L program. One of the promising is that we combine a lot of binders to see the optimization, and we are developing two E3L platform right now. And compared to certain breed CRM, we try to develop more compatible E3L. So E3L is overexpressed in immune cell. And so this is a target we are aiming for. So this is the end of my presentation. Thank you for your listening. And we will take a question from the floor. So let me start the question. So fortunately, PLK1, actually there's no approved PLK1 inhibitor or degradator. So I think your strategy is on the right direction, but at the same time, what is your competitiveness compared to those candidates in clinical trial? So it can reduce blood cell toxicity. To a nutshell, I do not show my data, but we compare onpasatinib, which is in clinical trial, but actually blood cell toxicity shows disparity. Our together is showing disparity in blood cell toxicity, and we have acquired that data. I am Ki Dong-hyun from Cyrus Therapeutics. And actually, E3 ligase is developed by your platform and at a single cell level. actually single cell specific E3 ligase expression level, and still actually there's a correlation between this and the actual function. Actually, you point out very well. Uh, Basically, we do single cell sequencing, but actually we do analysis at a single cell level. And on that note, correlation is observed. And E3 ligase, centering around the E3 ligase, which shows the correlation, we are developing those E3L platform. And another question? Further question? Thank you for your presentation. And PK/PD correlation is very important. So the candidate half-life stability is of importance. So when the protein is degraded and mRNA can create a target protein, so this will be remain in the cell line, and in cell line the stability of KD or half-life should be maintained at a certain level. So how can you maintain this? And second question is about, so actually could you please deliver your answer to the first question? Actually, when it comes to PD, the after the withdrawal of drug, 72 hours of degradation is witnessed. So this is the data we have acquired, and you have shown the in vivo data. TPD drug can be utilized as a first in class, or it should be utilized in a combination therapy. So what is your take on this? Is there synergy effect with the combination effect or monotherapy? And with the collaboration of Samsung Medical Center, and actually we are aiming for monotherapy as a first in class drug because there is no more therapy option here. So we first think about the utilization of our together as a monotherapy, but if necessary, we can utilize immunocancer therapy as a combination, ETO plus P or protein class and actually immunocancer therapy. We try to replace chemotherapy, or we are aiming for. We try to think about our possibility of replacing these three treatment options with our monotherapy option. If there's no question, I will actually F value is 20. Is it more than 20? So there's no PK data. So PK data is one of the biggest challenge for product. So actually, I have doubt because you don't show us the PK data. So how can you solve the PK data issue? But thank you very much for your thoughtful presentation. Actually, I want to answer my question because you said you are doubtful. We are now in the technological transfer. Actually, we are not able to show you this important data, which is very confidential. So based PK profile and distribution of small molecule is the data that we are showing. Thank you very much.
I would like to invite the next speaker from Inacure Therapeutics. We have Travis Williams, and he will make a presentation in English.
Hi, I'm Travis Williams, and I am the BD manager at Intercure Therapeutics. We are a small startup company that's focusing on TPD technologies here in Korea. And today I'm going to be talking to you about the overall trends that are happening in the TPD industry. I'll be going over a brief history of TPD. Then I'll talk a little bit about the current clinical trials, and then how companies are discovering their next TPD targets. And then we're going to switch gears a little bit, talk about some of the investments and deals in TPD technologies. And last, we'll finish with the future of TPD. So first of all, what is TPD? It's targeted protein degradation. And this example here on the screen is probably the most famous example of a TPD technology. It's PROTAC, And how protocs work is they bind two different proteins at the same time. The protoc on one side will bind E3 ligase, as you can see in green there. That is CRBN. And the other side of the protoc will bind your target protein. This is the protein that you want to have degraded. And once the protoc binds both the E3 ligase and your target protein, E2 will come along and ubiquitinate the target protein, and it will be sent to the proteasome and degraded into small amino acids. So how did we get here? Well, it first started in 2001 when Kathleen Sakamoto, Craig Cruz, and Ray Deshays, they took a covalent inhibitor of MET-AP2, added a linker to it, and added a peptide recruiter for an E3 ligase SCF. And they showed that their proof of concept worked. They got the first protoc in 2001. It wasn't until 2004 that another E3 ligase was recruited using the same method. They used a peptide to recruit VHL. And then for a long time, there weren't very many big developments, but in 2012, we got our first big breakthrough with a small molecule binder for VHL. It then took 3 of optimization of that until we got the first nanomolar degradation in 2015. Additionally, in 2015, it was discovered that the CRBN binders like thalidomide, lenalidomide, and pomalidomide can also be used as protocs and can also do nanomolar degradation. And it was with these 4 chemicals that the world of TPD exploded. So I have probably the most complete list of TPD companies in the world. I've done a lot of Googling, I've done a lot of data sheet searching, and currently there are over 75 global TPD companies that are mainly focusing on TPD technologies. Additionally, there are another 75 global companies that have realized the trend of TPD and have added TPD technologies as their pipelines. As you can see by the graph, most of these companies are coming out of the United States. But what may be surprising to some of you here is that China is also taking a huge interest in TPD technologies. There are currently over 35 Chinese companies working on TPD right now. And over there on the right side of the screen, Big Pharma has also been getting interested in TPD for a long time. Whether it's developing their own pipelines like Bristol Myers Squibb, or doing big licensing deals like Pfizer, or just partnering with these TPD companies to discover new TPD technologies for their targets, all of these big pharma are getting involved. So in these just 7 years after those first 4 chemicals were discovered, we've already seen a lot of companies enter into the clinical trials. Here are just the PROTAC clinical trials. There are 18 of them. Arvinas is the front runner with 2 of their pipelines already in phase 2. And I can't talk about all of these pipelines, but the overall trend is that most of their targets are pretty similar. They're mostly the androgen receptor, estrogen receptor, BRD9, IRAK4, or BTK. The second trend you'll notice is that almost all of their indications are the same. There is one exception, Kintor Pharmaceuticals. They are the first TPD company to develop a topical PROTAC in the world. And they're targeting the androgen receptor for alopecia, which is hair loss, and they're also using it for acne. So they're the first to really think outside of the box, or I guess outside of the body, with their topical cream. And then the last thing you'll notice is that almost all of the E3 ligases that are in these clinical trials are pretty much the same. They're all CRBN, and they're all VHL. Next, we have the molecular glues. And one of the first things you'll notice about molecular glues is, again, they're all CRBN as their E3 ligase. And because they're using CRBN as their E3 ligase, a lot of the molecular glues are highly target restricted. There are currently only about 5 targets molecular glues are able to target with these CRBN binders. They're the IKZF1, 2, and 3, GSPT1, and CK1 alpha. And then you also notice that a lot of these are targeting all blood cancers. So how do these TPD companies discover their own targets? Well, if you go to a lot of PROTAC websites, almost all of them will talk about undruggable targets. So what is undruggability? Well, for an inhibitor to work, you usually need to bind to the enzymatic site of a protein or to an allosteric site of the protein. However, PROTAC can bind anywhere on the protein as long as that protein gets ubiquitinated. And that's one of the big advantages of PROTACs and TPD technologies. But the question is, do you need undruggable targets? And I would say no. Most of the PROTACs in clinical trials are all druggable targets. So these are the other things that you should consider when you find a new TPD target. For the protein aggregates, the gene amplifications, and the protein overexpression, inhibitors don't work well against all of those. So definitely PROTACs have an advantage there. For the scaffolding functions, inhibitors, they will just bind the protein. They don't remove the protein. So a lot of the cancer pathways can still interact with that protein and still function. PROTACs can definitely have an advantage there. The isoform expression, you can look for proteins that have mutations elsewhere on the protein other than the active site. And then here's where I think PROTACs have a huge advantage over inhibitors, is that they are better at resistance mutations. For inhibitors to work, they have to stay bound to the target all day. If they fall out, the protein works again, or until the next dose comes. So if a mutation happens at the inhibitor site, you can lose function of that inhibitor. However, a PROTAC only needs to stay bound to the protein long enough to have the protein ubiquitinated. So I think there's a big advantage of PROTACs there. And finally, for protein localization, inhibitors go everywhere in the body. You really have no control of where they go. So if you use a specific E3 ligase for a specific tissue, you can control the protein localization. So let's say you found your target. And how do you find a binder for your new target? Again, if you go to all the PROTAC companies, over half of them won't tell you how they're screening for their binders. The other half will tell you, and they're using almost every method available to find binders for these proteins. A lot of them are doing high throughput screening or fragment screening. And then a lot of companies also have their own libraries to do low throughput screening. And then also a lot of companies are doing in silico or AI screening of their targets. And some companies are even writing their own code for machine learning and for computer modeling. But by far still the most popular way to find a binder for a TPD technology is with DNA encoded libraries, DEL. And there are 2 ways to do this. You can have your own in-house DEL screening system like what Nurix does or what Plexium does, or you can just outsource it to companies like HitGen, Vibergen, or XChem, which is what a lot of TPD companies are doing. So at InterCure Therapeutics, we use several of these different methods to find our binders. Once we decide on a target protein, we'll go ahead and do a computer-assisted virtual screening of that target. We'll find the pockets and the binders for that, and then we will AI chemically optimize it until we feel satisfied and can synthesize the chemical. Once the chemical is synthesized, we will then test it in cells to make sure that the target protein is being degraded. And we will continue to manually optimize that chemical until we have a viable drug. All right. Oh, one thing I did not talk about is down there at the bottom is the other side of Protox, which I think a lot of companies are ignoring. Most companies will just use thymaldamide or pomalidomide, put a linker on it, and then find a binder. But at InterCure Therapeutics, we spend a lot of time optimizing the E3 ligase of the PROTOC as well. And if you do this, you'll be able to get enhanced CRBN binding. You'll be able to improve your degradation and inhibition of these proteins. And you'll get enhanced selectivity, which also always increases its safety profile. So we think you need to optimize really both sides of the PROTOC to actually get it to work eventually in a clinical trial. Next, we're going to switch gears, and we're going to talk about some of the venture capital interest in TPD companies. And there are currently 13 companies out there that have received over $100 million in financing from venture capitals. Most of the early PROTOC companies that have now a clinical trial pipeline, most of them have IPO'd by now. Some of the newer companies like Plexium or Montathera or Triana or Neomorph, they are all focusing on molecular glues over here. And other companies like Frontier Medicines and Chasma Therapeutics, they're focusing on a different type of degrader technology, the autophagy degradation. And additionally, there's Lycia Therapeutics on there as well. It's not just the VC companies that are interested in these TPD companies, it's also big pharma. And if you look at the deals that have happened since 2020, there have already been over 20 deals. And most of these deals from big pharma have been into the area of molecular glues. They're partnering with these companies and they're using their platform technology to help discover new molecular glues for new targets. As you can see, also PROTOCs are still quite popular and they're still generating lots of deals and interest in the industry. But one of the new trends is that there's been deals in these CNS targets as well. And again, at InterCure Therapeutics, we're working on a method to be able to target these CNS targets. And our method to do that is UpTech. It's an autophagy targeting chimera. It works very similar to PROTOC, and we bind the target protein. However, on the other side, instead of binding an E3 ligase, we bind the autophagosome tethering proteins. Once this complex is formed, it will be pulled into the autophagosome, and when the lysosome merges with this, all of the proteins inside of it will be degraded. So this technology works really well for large aggregate proteins, or can even be used on organelles inside the cell like mitochondria. And we think this technology has great potential if we can get it to work. And we think the first company that succeeds in being able to get this technology to be BBB penetrate will have a huge advantage in the entire industry. Next, I'm going to spend just a little bit of time talking about the other types of degradator technology. There's the first technology here, DubTex, or DUBs. DUBs are deubiquitinating peptidases. And these aim to take off the ubiquitin from your targeted protein. And by doing this, it saves the protein. One company that is working on this right now is Stablix Therapeutics. Next, the other degrader technology is the antibody degrader conjugate. It works just like an antibody drug conjugate, but instead of the drug or warhead, you just replace it with a degrader. And this technology works great because you can target your degrader to the specific tissue that you want with the antibody, and then the degrader will then go inside through the endosome and degrade the target protein in your targeted tissue. So it's quite safe. And one company working on this is Aurum Therapeutics. The next technology down there, Litax, this is another antibody technology. And what this technology does is it combines an antibody with an oligoglycopeptide. So the antibody will target the surface membrane protein or a circulating antibody, and the oligoglycopeptide will bind to the mannose-6-phosphate receptor. Once it does this, it will be pulled into the cell and degraded in the endosome. And lastly, a couple of the other technologies are the ribotax. They bind RNA, and they connect the other side of the ribotax to an RNase so that you can degrade RNA. These are still very early academic technologies, but there's a lot of excitement for them. And then lastly, there are the phosphorylation tacs. These are a kind of technology that's coming out of Craig Cruz's lab, and they connect phosphatases or dephosphatases to your target protein so you can turn on or off your technology, or you turn on or off your protein. And then last one there is the ATAC, and that basically is just another way to target the target protein to the autophagy degradation system. So a lot of these technologies are still in the very early stage, but if you remember, PROTOC was also in the very early stage 20 years ago. And right now we're already seeing lots of PROTOCs and lots of molecular glues in clinical trials. So I look forward to seeing some of these new technologies in the clinical trials sometime in the future as well. And overall, for the trend of TPD, I think it will continue that molecular glues will be popular as long as you can find new targets for them. And PROTOCs will also be popular as well if you can continue to optimize them and make them smaller. And there will also be a huge interest in CNS targets as well, and using some of the autophagy type of degradation for those. So thank you.
제가 먼저 질문하겠습니다.
I would like to ask our first question.
Yep.
오늘 BD 관점에서
Today, as for the BD,
이노큐어 프로탁 얘기를 안 했는데
you didn't mention the inocure protoc.
타겟 몰리큘 중에서
So among the inocure protoc
매력
molecule, which one is the most fascinating one?
I think we can talk about that. I think we can talk about our pipelines. We have one specific PROTAC that we're developing for the BET target out there. It works, it's currently a pan-BET degrader and can be used for lots of different solid tumors, and of course for the very high unmet needs cancer of nut midline carcinoma, which is what we're developing for. Yeah.
예, 질문. 한국말로 질문하세요.
You can ask a question in Korean.
Jeong-Lim from TheraPEX. I wonder if you have a particular in-house program to approach the linker for optimization.
If I have a what program?
For in-house protocol or program to tackle the optimization of the
Right.
linker.
So what we do at Innocure Therapeutics is not only do we design the binders and design the E3 ligase binders as well, but we have our own in-house chemistry team at Innocure Therapeutics that is very experienced, and they will work to optimize the linker as well. So of course you want the linker as small as possible, but sometimes you need it a little bit longer just to overcome the ternary complexes that are happening in these proteins. You would have to talk to our chemistry team on that. They have a lot of expertise and they do a lot of computer 3D modeling to figure out the size of the linker and exactly which functional groups need to go into these protocs.
Is there any other question? If not, thank you very much.
저희가 처음 세
And
턱은
actually, the three talks are about target protein degradation. And the last presenter will deliver on the different type target protein upregulation. So why don't we change our gear? So preparing for this session, this is the last listening to target protein upregulation. This is why we invite Park Seung-bom, CEO and founder of Spark Bio. Thank you for your kind introduction. Once again, thank you for inviting me to this important event. And this session is very specialized session on degradation, but let me switch the gear. So I would like to touch upon updegradation. And when it comes to the presentation, I will briefly talk about the overview and our strategy. And actually, for 20 years, I have taught at Department Chemistry, Seoul National University. And I am thinking about the upregulation and as a researcher. And I would like to think about how to develop drugs and then how can we commercialize this drug. And we invite Choi Young-il, our CSO, and Kim Young-ho, our CFO, are invited. We were established in 2016. We are a spin-off company for SNU, and actually we have 35 employers, and we successfully carried out $57 billion one. And actually, we are trying to develop first-in-class small molecule drug. So in order to do so, we try to develop new platforms and how can we optimize the development of small molecules. Through these molecules, we are studying on PPI molecule modulation. And also we try to utilize phenotypic assay to realize this through screening. So how can we visualize this based on image-based screening? And then we also need target ID technology, and this is co-technology platform that we have. And this platform are born out of Seoul National University's lab. And actually, students are engaging on innovation. And in order for us to develop this idea, Spark Biopharma is trying to commercializing this. And we identified candidate out of lead, and we will carry out preclinical and then clinical phase 1. So we bring this innovation into our reality. And this is a scientific asset creation process. This is why the platform is very important. And also, we have to increase the value by licensing those candidates that are whose MOAs are not known. And so we have a lot of talked about the target protein degradation. So in terms of cancer immunotherapy or oncology, actually, there are 3 pillar treatments: surgery, radiotherapy, and chemotherapy are of importance. In the past, however, these days, immunotherapy is a first-line treatment. In 2018, after the Nobel Prize, and the estimated percent of patients eligible for checkpoint inhibitor drugs are increasing. And actually, immunotherapy will be used as a first-line therapy increasingly. But the problem here is that actually the response of the immunotherapy is very low, and efficacy is very good, but this is not good depending on patients. So immune checkpoint inhibitors should identify those masked tumor cell. However, when the immune response is not observed, is useless. So cold tumor environment is what is about immune suppressed environment. So the many researchers are focusing on turning cold tumor into hot tumor, so to sensitize immune cell. And there are a lot of strategies here, but as I mentioned before, immune supply system is cold tumor. So how to boost our immune response? So in order to boost immune system, we have utilized a lot of CAR T cells or chimeras on immune cell to boost the immune response. So this is all about turning cold tumor into hot tumors, of which the sting is the front runner. And so sting is detecting cytosolic dsDNA, and actually these serve as an alarming system to our body. So this is a sensor molecule sense the intrusion of cytosolic dsDNA, and through ligand, sting was delivered. This was delivered to the sting, and TBK pathway and interferon gamma and interleukin 6 are boosting the immune system. This is good for immune system. So cyclic CGGMP activates the intruded cells. So the sting can hijack this, and on the same mechanism of action, it can boost the immune system. So small molecule-like sting agonist is under development. 22 companies are doing clinical trials, and major pharmas are all jumping in this market, and there are a lot of licensing deals. And clinical phase 1 and 2 are carrying out, and this is about IT or IV cancer injection. And when you see the structure, and actually there is a synthesis of structure, or there is a materials that whose structure is not revealed. So through this test, there are efficacy in vivo and in vitro. However, in clinical trial setting, there is no good efficacy observed because there is a lower efficacy, or there is only partial response in phase 1. So many people are thinking about the PK of these materials are not good, or instability or affinity might be a problem. So actually modulating sting is important. Development of sting agonist is not competitive, so we try to change our approach. So we are starting to think out of the box by utilizing other approach to modulating sting. Actually, sting was activated by cytosolic GMP, and this leads to ubiquitination, and which will be degraded. However, sting without activation can be ubiquitinated, remaining as a dormant in TRIM29. So sting agonist, when this is injected, there is a little sting activity here. So this is why there is a little efficacy in clinical trials. So TRIM29 can be inhibited, which leads to overexpression of sting. So when we inhibit TRIM29, there is a metastasis. So actually, direct TRIM29 elimination promotes metastasis. So this is a undruggable target to modulate proteins by inhibiting degradation. So first thing, we can inhibit degradation. So through proteasome inhibitor drug, we can inhibit degradation. And this is something that is unimaginable. So we can target more sensitized cancer cell to be dead. And actually, we can induce degradation by protect molecular glue, and this is research area on undruggable target, and this trend will be ongoing. And we can E3 ligase cannot ubiquitinate distant target protein, so PROTEC brings E3 ligase and target protein into close proximity, which will be degraded. And the market is on a constant growth. And there are a lot of sessions, and actually I have a presentation in AACR, and the discovery on E3L is something that we are seeing. And also BZ's target, actually down regulation of target is causing proteins is important. However, we have to upregulate disease fighting proteins. And actually, we have to prevent the degradation of disease fighting proteins. So upregulation of disease fighting proteins can be of a good approach. So there is a target protein, and this will be ubiquitinated via E3 ubiquitin ligase, but this is problem, so we can block the PPI between the protein of interest and its E3 ubiquitin ligase, thereby upregulating the disease-fighting protein. So UPRIS. This is the meaning of resurrection in Iceland world. So UPRIS can allow us to upregulate the wanted protein. So we have tuner interaction, right, in protein. However, we can actually UPRIS can block the PPI between the POI and its E3 ubiquitin ligase, so which is very easier for us to develop. So PROTAC is trying to degrade target disease-causing protein. However, UPLIS is to upregulate disease-fighting proteins. This is all about complementary thing. So the STING protein still is degraded by E3 ligase. So this STING activity is degraded on E3 ligase, which leads to limited immune response. So we can develop a small molecule to limit the function of E3 ligase, which leads to upregulation of disease-fighting proteins. How can we test this? TRIM29 and STING proteins interaction inhibitor will be discovered. So we can estimate which interaction would be happening, but there's a TRIM on STING, and their close proximity are leads to fluorescence, and STING and TRIM, entomcytum and lasbis are tagged and 2x2x2 matrix. So 8 pairs are possible. So 8 pairs data show that a certain pair is more fluorescent, and their fluorescence will be disappear. So small molecule inhibition leads to loss of fluorescence, and this means this leads to blockage of PPI interaction. The fluorescence is disappearing, and there are new materials that affect this interaction. So the basic concept, the amount of STING, E3 ligase, TRIM29 is modulating this. And we can increase the TRIM29, which leads to downgrade of STING. And SI-TRIM, STING-TRIM is inverse correlation. So there there is a stable amount of trim, but the sting is degraded by our SB24011. So stream 21 induced Ka48 linguist-specific ubiquitation of sting is what we are studying. So we only leave K48 and there is a specific event, a selective event, and we can identify the inhibition effect. So there is no effect when we do test, but texamide, which inhibit translation, ectomycin, which inhibit transcription. But even in these circumstances, we do witness the efficacy. So we do not control transcription or translation. Actually, we inhibit the degradation, thereby increasing the disease target, fighting target proteins. So the TRIM and STING interaction. So our molecule binds STING, and SESA, IPVR, LDL4 are something that we can observe this effect. And we have obtained co-crystal structure. So you might know this. STING is a homodimer form, and active site is here, and all the STING agonists are binding to this site. So our molecule is not compatible to STING. So this leads to a rational design. So we identify the modulation of activity here. And after that, the amount of STING is increasing according to FEX and WESTA with administration of our molecule, but there is no change in mRNA level, so there is no effect on transcription and translation. So there is an increase in amount of STING. There might be lead to greater immune response. And there are interferon beta, IL-6, IL-15, and cytokine production are boosting in a synergistic manner and time dependently and dose dependently. So let me rush. So we have identified the in vivo efficacy. So we do IT. Sting systematic increase is problematic. So we do IP administration, and we do IT administration. So 1 microgram, 3 microgram will be injected, and CT26 inoculation after that, compared to cyclic GMP, and this is the data result. And the red line is not that effective, but STING agonist, when this is combined, there is a good synergistic effect. And there is no body weight change. And when we do administer our molecule, NDSC decreases, T cell increases, and of course, perfin, interferon gamma, which increases the T cell fitness, increases. Might be a macrophage or M1, M2, and macrophage immune-related cell show increased level of STING. So STING agonist can create a synergy effect with our small molecule. However, this effect should be observed in primary tumor, but also in the distal tumor. So before that, sitting golden gate mitigation shows that without STING, we do our molecule administration, there is no effect. So this is all about this is dependent on STING, and 1 microgram and 3 microgram. With this combination, we have witnessed a tumor volume decrease in primary tumor and also in the distal tumor size decrease, which means through IP, this can be injected to the primary tumor, but the training T cell by primary tumor, and this is circulated through our body, and also the distal tumor size is decreasing. Then, which means our molecule can be applicable to the other metastasis tumor or distal tumor. And cyclic GNP STING agonist is in clinical trial. PD-1 can be used as a first care, so we do combination with PD-1. And the blue one is PD-1. And CT26 cancer is not respond to anti-PD-1, but our molecule is well responded to CT26 cancer. So our molecule is working well in combination with immune checkpoint inhibitor, especially PD-1. And there is a similar immune cell behavior observed. So increase in STING can lead to anti-cancer efficacy. And our molecule through Omni strategy is firstly carried out and developed. And SB24011 is a lead candidate, and another candidate is in preclinical trial. So we try to boost the STING activity by upregulating disease fight proteins, and this shows a strong anti-cancer activity. And also there is a good possibility of combination with immune checkpoint inhibitor. And we are planning to file IND in Korea, and SBP145 is in preclinical trial, and we are aiming to filing an IND in the 2024, and we will get into the phase 1 in that year. So the upregulator of STING can lead to T cell memory. As I mentioned before, increase of STING in T cell level is witnessed, and prevent interferon gamma and T cell fitness also are increased. And I delete the data intentionally, but in MHC1 loading OBA1 through this, there is a specific antigen training, and this leads to over one expression cell. So memory effect train T cell, and which increase the immune response. And another question? Thank you for your thought-provoking presentation. I am a Busan University College student, and SB compound is what you found, but actually electrocycle combination is not out of sudden. So there are a lot of drug candidates among them. I want to know the procedure of discovery and selection. And actually the basic concept that we have is that target protein degradation is very rational. And of course, the limitation in rational approaches, very similar approaches. So PPI interaction blockage or modulating. Actually, I believe POI and E3 ligase interaction blockage might be possible. This is what we have thought, and we do have IP here. So phenotypic cell-based SL evaluation shows us a lot of hits, and of which, rather than TRIM, we try to identify molecule that is not competitive to STING, but also binds to STING. So there might be another approach that if we do change our direction, but the behavior that we want to see, and also we can modulate another function of STING because our molecule is binding to the STING. And there is another type of UPNIS project through which PPI modulation based on regulation of protein might be possible. This might be an example, and we are planning to develop platform building on this data. And another question? Thank you for your presentation. I am not authority on this field, and so turning a cold tumor into a hot tumor is of great importance. So this is a goal that you would like to achieve by STING? Yes, that's right. So when we do make genoleft and the cold tumor can be turned into hot tumor, so there is a visibility or evidence that we can see. There is a specific marker here. Immune. So blood chemistry is a method to see the activity of immune response. So the tumor microenvironment is suppressing immune system. This is well known to us, but we try to see the increase of infiltrated T cell and other immune cells. TMS1, a vehicle treatment. Compared to this, we can identify the mechanism of action of our molecule. So tumor environment should, the change of tumor microenvironment is what we are aiming at without in non-invasive manner. So we conclude the session. So thank you all for taking time and listening to this presentation, and most of all for presenters. Thank you, and your presentation showed the global competitiveness of that your companies have, and I really do good at science intelligence, which is why I invite these prominent speakers. So science intelligence writer, as this writer, I would like to write an article based on takeaways of this session. Thank you.